Okay. In this lesson, we will discuss some differences and similarities between the prokaryotic cells and eukaryotic cells. As we have discussed prokaryotic cells and eukaryotic cells in detail, as a summary, now it is easy to compare the prokaryotic cells and eukaryotic cells. The first thing we compare between the prokaryotic and eukaryotic cells is their size. Prokaryotic cells have a diameter from 1 micrometer to generally 3 micrometer, whereas the eukaryotic cells have a range of their sizes. Animal cells have a diameter up to 20 micrometer, whereas the plant cells have diameter up to 40 micrometer. Prokaryotic are always unicellular, whereas the eukaryotic cells may be unicelled or maybe multicelled. Cell wall is always present in peptidoglycans and in prokaryotic cells, and it is made up of peptidoglycans. Whereas in eukaryotic cells, the cell wall may or may not be present. In animal cells, cell wall is, I have written this in the column of nucleus. So I just, this is cell wall with an arrow. So cell wall may be present in eukaryotic cells or may not be present in eukaryotic cells. Animal cells don't have cell wall, whereas in plant cells, if plant cells, the cell wall is present and it is made up of cellulose. Okay, and in animal cells, it is not present. Okay. DNA. Prokaryotic cells have circular DNA, circular and naked DNA, whereas eukaryotic cells have linear DNA, and it is associated with histone proteins. So it is not a naked DNA. Ribosomes present in prokaryotic cells are smaller and they are called 70S ribosomes, whereas in eukaryotic cells, 80S ribosomes are found in cytoplasm, whereas 70S ribosomes are found in chloroplast and mitochondria, as the chloroplast and mitochondria are also found in eukaryotic cells. So you can say that 70S and 80S both are present in eukaryotic cell. Membrane-bound cell organelles are absent in prokaryotic cells, whereas they are present in eukaryotic cells like chloroplast and mitochondria. They are not found in prokaryotic cells, whereas they are present in eukaryotic cells, mitochondria and chloroplast. Chloroplast are present in eukaryotic cells. Cell division, there are two types of cell division involved in asexual reproduction and in growth of organisms. In bacteria, it is known as binary fission. Details of the binary fission are not in our syllabus. However, in eukaryotic cells, the cell division is mitosis, and we will discuss mitosis in detail in another lesson. An example of bacteria, an example of prokaryotic cell are bacteria, whereas examples of eukaryotic cells include animal cells and plant cells that I have already mentioned here in the first row. Nucleus is absent in prokaryotic cells, and double membrane bound nucleus is present in double membrane bound, double membrane bound nucleus is present in eukaryotic cells. So that's about the differences between prokaryotic and eukaryotic cells. Now we will discuss some questions on animal cells, plant cells, and the differences in between prokaryotic and eukaryotic cells. So let's see. Let's see, this is an animal cell. The different animal cells have different shape and different sizes, and they may have one or the more component more abundant or less abundant depending upon their role in the living organisms. This is a ciliated epithelial cell, and it has cilia present here. So these are cilia, and this is B a cell membrane. Okay, we have these mitochondria, and this is the nucleolus. Okay, nucleolus, not the nucleus. It is nucleolus. Okay, this whole structure from here to here is this is nucleus here. So this is nucleus. Okay, this cell you can see has an abundant number of mitochondria. These are all mitochondria present in it, as it has to sweep the mucus with the help of movement of sweeping movements of these cilia. So it needs energy for that, and to provide ATP, there is a large number of mitochondria in them. As these cells are not secretory, so any structures involved in producing the secretory proteins are less abundant in this cell. Okay, we also have this this part. This is maybe nuclear envelope, and we have this chromatin, and we have this nuclear pore, and we have these. See, this is slightly different view as we generally draw the Golgi bodies. These are flattened sacs called cisterna, which are stacked one above other. And generally, when we draw the structure of a Golgi body, that is, these the cisternae are clearly distinguished from each other, and they may have the lysosomes, pinched off from the ends of these cisternae. Here, it is though it is difficult to identify, but yes, this is the Golgi bodies or Golgi apparatus. Okay, so let's see what are the questions on this cell or on the different parts of this cell. Here we have complete the table below by writing the appropriate letter from figure 1.1 to indicate the structure that carries out each of the functions listed. The first one has been completed for you. Facilitated diffusion of glucose. Obviously, the channel proteins are found in the cell surface membrane, and the diffusion of glucose, facilitated diffusion of glucose takes place through the cell surface membrane. So B is cell surface membrane. Creates current to move mucus, so that should be cilia. And you can identify with the letter from the diagram. Then aerobic respiration, this takes place in mitochondria. You can again identify the mitochondria with the letter from that diagram. Makes ribosomes, it is nucleolus. This is involved in formation of ribosomes. You can identify with the letter from the diagram. Site of transcription, this takes place in the nucleus where chromatin is present. So nucleus or chromatin, you can identify nucleus or chromatin for transcription and packaging proteins into lysosome. This is the Golgi bodies or Golgi apparatus. You can identify Golgi apparatus with a letter from the diagram. So that's about this animal cell. its different roles of the different structures and identification of the different structures. So let's see, we have another animal cell. In the immune system, a plasma cell develops from an activated B lymphocyte. Mature plasma cells synthesize and secrete antibodies. Figure 4.1 is a diagram of a transmission electron micrograph of a plasma cell. You can see here, this cell has an abundance of, these RER, or rough endoplasmic reticulum. But in the ciliated cell, you have seen endoplasmic reticulum, or rough endoplasmic reticulum was less abundant. The reason is the different role of the ciliated cells and the plasma cells. Plasma cells are secretory in their function, so they need a lot of antibodies to be made, a lot of proteins to be made, so that they are secreted out of the cell. So that's why for synthesis of larger amount of these secretory proteins, they should have abundance of rough endoplasmic reticulum in them. So this is rough endoplasmic reticulum. These are mitochondria. Okay, we have these Golgi apparatus or Golgi complex. We have this whole structure. This is nucleus, and I don't see any smooth endoplasmic reticulum here, but yes, we can identify there are no other structures seen here, no centrioles visible in this structure. It doesn't mean that the centrioles are not present in animal cells. Centrioles are found in animal cells, but in this cell, it's not visible. It's not shown in this cell. However, these Golgi vesicles are shown in this cell, right? Ribosomes are also visible on the RER, or the rough endoplasmic reticulum. So let's see the question on it. The plasma cell can be seen in greater detail using an electron microscope compared with using a light microscope. Describe the extra details of the nucleus that can be seen using an electron microscope. Under an electron microscope, nuclear pores are seen, a nuclear envelope or two, nuclear membranes are seen, and RER, continuous from outer nuclear membrane is also visible. Ribosomes and the outer nuclear membrane are also visible. So these are some structures that the extra details that could be seen under electron microscope. You will have these questions in these sheets with the mark schemes with them. These are carefully selected questions with their mark schemes, and that would help you to understand these concepts and prepare your notes. Okay. And then explain why cell structures such as ribosomes and rough and smooth endoplasmic reticulum cannot be seen using a light microscope. So very simply, you should write that light microscope has low resolution. You can write it is 200 to 300 nanometer. Okay, and these structures, you can write the structures of these sizes of these structures, like ribosomes are 25 nanometer, and rough endoplasmic reticulum and smooth endoplasmic reticulum, they have cisternae that they have smaller thickness or structures than this resolution. So that is why they are not visible under light microscope, right? So this is another plasma cell. This is a transmission electron micrograph of a plasma cell. The similar structures are seen, but you see, this is a drawing of the plasma cell, which shows the structures with more clarity, and you can easily identify them. But sometimes it becomes a bit difficult to, identify the structures in, the images. electron photomicrographs or photo micrographs. So this is, the rough endoplasmic reticulum here. You can see these are the mitochondria here, and this is chromatin present inside, and this whole structure, this is the nucleus with the nuclear membranes, nuclear envelope, and more details are not visible, like Golgi bodies and the lysosomes, ribosomes, that are not visible. If we, enlarge or zoom in this structure, maybe we can find the ribosomes attached on the rough endoplasmic reticulum. Okay, so let's see what are questions on this topic. You have to identify, write the name of the structure, okay, and functions of the structures within the plasma cell. So see, if A is a nucleus, you know the functions of the nucleus. It is involved in protein synthesis. Transcription takes place in it. mRNA is made, and that genetic information is passed from the DNA to, the ribosomes through the mRNA. And then B is, what is the B? Yes, B is mitochondria. So it is an ATP formation by aerobic respiration, okay, which is needed for, protein synthesis in the plasma cells. And C is, what is the C? C is rough endoplasmic reticulum. So obviously rough endoplasmic reticulum is needed for formation of proteins, or you might write translation. So B is mitochondria, and it is nucleus. So it has genes for protein synthesis, and RER, protein formation. So that's about animal cells. We will continue in the next class and discuss some more questions on plant cells, the components of plant cells, and then we will have some more questions on differences between prokaryotic and eukaryotic cells. So that's it for today. See you in the next class, inshallah. Thank you.